Okay. People still wander in, but I'm going to start introducing our speaker today, Paul Koch. Koch?
Yep, gosh.
Koch, from University of Santa Cruz. And Paul gave a really interesting talk yesterday about something that yeah, mega herbivores and their gut biome and the amino acids in their bones. In Africa, modern, and Pleistocene megafauna in North America. And he somehow wrapped it all together. And
Don't
yeah,
pronounce.
yeah. So we'll see what he can do today. Paul comes from New York. Um,
Jersey. Jersey.
oh, okay. And got his undergrad at University of Rochester, his PhD and master's from University of Michigan. And then I didn't connect all the dots until how you got to UC Santa Cruz. He recently stepped down as dean and has joined the common faculty again. And is going to, start up all these research projects he had to put on hold. And I've just heard this is going to be the first one to be written up.
Yeah, I'll say it.
Okay, he'll tell us more about that. But without further ado, Paul.
Thank you. Thanks. It's been great visiting. My first time in Logan, first time at USU, and I spent a lot of time in Utah, but first time up here, and it's a great community. I have to say, it's a really diverse, and I've talked to a wide range of people in my time here, and it's been really great. So today, I'm going to talk about sort of. I was asked by some of the students to talk about kind of how I do research and like how I find my questions. And this is one part of the work that I do, which is using the fossils of living species to do conservation or try to get information that's useful for conservation planning. Um, and I'm going to tell you two stories. One is going to be about lemurs, and the other is going to be about seals, Antarctic seals. And they were both done in my lab. The first project by grad student Brooke Crowley, and the second project mostly by me, but with grad student Emily Brault from in the Ocean Science Department. So, seals. But the first thing, come on, as a paleontologist, um, I imagine the world the way it was for most of its history, and that's not the way it is. And it's important to realize that the world that we study now isn't the world that existed for 10,000 years ago to 20 million years ago. So this is the distribution of mammal biomass on Earth today. Um, that's us. That's our domesticates. Cats and dogs together equal the biomass of all wild land mammals. And marine mammals, about twice that amount. So it's almost all the food on Earth is either on the hoof as cows or as us, right? There's very little space left for wild stuff, at least wild mammals. So that's sobering. Um, it was different in the past. This is 25,000 years ago in South America, and sloths showed up yesterday. That was fun. I told people yesterday they're not as weird as hippos, and I'm sticking with that. I wish I had samples of this animal because it might be as weird as a hippo. But estimates of biomass prior to the loss of all these large animals let's say prior to the loss of all these large animals, about 120 megatons, whatever these units are. I think they're like kilotons. I don't know. Anyway, 5.5 times greater biomass of mammals 10,000 years ago than of wild mammals 10,000 years ago than there are wild mammals today. So it's actually pretty remarkable. Come on. to think about how it is that, what was it, 140 MTs of wild mammal biomass 10,000 years ago, 10 times that biomass today, most of it as, humans and the stuff that we like. Like, how does that happen? How do we, like, where does that come from? Like, we're made of things, we're made of proteins. I talked about this yesterday. We're made of proteins and fats and minerals. Like, where does it all come from? And it's important to remember that, a lot of it comes from fossil fuels, both fossil fuels used to do work to extract these resources, but literally the production of turning the N2 gas in the atmosphere into ammonia, which ends up as fertilizer on the fields. Like half the nitrogen atoms in your body are made in reactors like this. And if these reactors stopped working, we would have issues. Okay, there'd be some issues. So, and not only are they run by fossil fuels, but actually ammonia is part of the chemistry here. So you need, sorry, not ammonia, methane is part of the chemistry here. So you need fossil fuel extraction to drive this in addition to do the chemistry. So, that's where ultimately the protein comes from, and the calories come from the Green Revolution, right? From the work done by scientists around the world to increase the production of food and to mechanize it so that we can deliver maximum calories per plowed field. And that's how we can have 10 times the biomass of the mammal biomass of the natural world. It's a remarkable feat of engineering, and one I hope we can continue to juggle for centuries to come, or can juggle as we reduce the size of the human population. So we live in a really different world, and a world that's shaped by forces nothing like what those Pleistocene megafauna experienced. And the other thing to remember about the past is that it wasn't always like this temperature-wise. So this part of the curve, this is a CO2 curve that goes back to 65 million years ago. Don's advisor kind of co-led the team that put this curve together. It was most of his career assembling these data. These are data on temperature measured using records from tiny little fossils at the bottom of the ocean floor. And you can see that, there was a period when the Earth was very warm. I've done a lot of work on that point right there, the warmest period of the last 65 million years, as has Don. But, the Earth has been dropping in temperature this is when the dinosaurs checked out 65 million years ago. There was a hothouse world here for the first, 25 million years. There began to be cooling. And then over the last, note there's breaks here in the timeline, right? So we switched to 5 millions of years here, and then hundreds of thousands of years here, and then tens of thousands of years here, and then tens of years here. So the timeline is getting, faster and faster and faster, shorter and shorter and shorter as we move across this diagram. But what you can see is that, for humans, modern humans appeared about between 600 and 400,000 years ago. And agriculture, appeared around here in one of these quiet spells relative to the noisiness of the climate record. And, we're projected to go to places, depending on which of these curves we follow. We could go back to the Eocene or, one of these time periods climate-wise that we haven't been to for tens and tens of millions of years. But this record of climate change and of different worlds, both different worlds with respect to the way the biosphere worked and different worlds with respect to the climate of those worlds, gives us a chance to look at how animals might have behaved. I care most about animals, how animals might have behaved. The animals that are alive today might have behaved under different conditions. And that's what I'm going to focus on today, which is, if we're going to be worrying about the vulnerability of species to anthropogenic change in many of its manifestations. We need to worry about whether the organisms are exposed to change, or in a place where there's something happening. That's pretty much everywhere now, but more or less. Are they sensitive to what's happening, or do they not care about it? And then what capacity do they have to respond to that change? What adaptive capacity do they have? And the options, if exposed and sensitive, are the four following. Well, sorry, the options are be insensitive and stay in place. Okay, that's great. You don't care. Evolve or respond plastically. So change who you are. Move if there's any place you can go in time, or go extinct. Those are you got a suite of responses. And what I try to do in this type of research, where I'm using the fossils of living species, is I try to study how sensitive they are and how they might change. Right? How might they change? Are there things that they did in the past that we've never seen them do today? And would that be an avenue that we might imagine them, or that we might assist them in reaching through interventions today? The main way I work on this is using isotope chemistry. Yesterday, I didn't talk at all about what isotopes were. I'm just going to say, versions of the same chemical element that have different neutrons in them, that give them slightly different masses, and those differences in masses let them be sorted by natural processes. And sometimes that sorting creates labels that we can just track the flow of mass around. And sometimes that sorting is sensitive to the rate of important processes. So we can track the extent of the activity of processes, like, I don't know, the limits that water availability impose on photosynthesis. So you can study process and how that's different in time, physiological processes sometimes in animals, and you can trace the mass fluxes across time and space. And the first example of this I'm going to talk about, I mentioned lemurs. So come on. So this is Madagascar 4,000 years ago. Hippos, the giant flightless birds, elephant birds, lemurs of various size, various varieties. The biggest lemur was the size of a gorilla. And that's not the world of today, of course. And this is Brooke Crowley, the student who came to work with me on mountain uplift, and then one day said, mountains are cool, but I really like lemurs. And for my PhD, I'm going to work on lemurs. So she did a master's on the uplift of the Sierra and a PhD on lemur ecology. Go figure. So you can tell right away that the lemurs of the past are different from the lemurs of the future, the lemurs of the present. These are some limb bones of extinct lemurs. These are the extant lemurs. I think the largest one now is 5 kilos. They were, I said, gorilla-sized in the past. And their extinction began about 2,000 years ago, after the arrival of humans onto the continent. And in the arid southwest of Madagascar, they hung on till about 500 years ago. And I'm going to show you data from that arid southwest, sort of the last stand of the big lemurs in Madagascar. And just a little bit about the ecology. Today, so I'm going to show you data from fossil lemurs that are at these spots with circles, as well as data from a modern reserve that's here along this river. And there is one fossil site that's close to that modern reserve. But most of the other fossil sites are on the coast. And the habitat is a mix of this spiny scrubland, and then right next to the rivers, there is this more dense riparian vegetation. And the highest concentration of lemurs today is in those riparian corridors. And that's where the reserves are. But the spiny scrub makes up the bulk of the territory, and the lemurs are rare and decreasing out there, and those aren't protected areas for them. The protected areas are in the riparian corridor. So I'm going to show you plots like this where there's carbon isotope data on the x-axis and nitrogen isotope data on the y-axis. The carbon data, basically, the higher the number, the more of the rare carbon isotope 13. The lower the number, the less rare carbon 13 there is. For nitrogen, it's the same story. The lower the number, more rare nitrogen 15. The higher the number, more of the rare nitrogen 15. And I'm not going to get into the mechanics of why these things I'm going to say next are true. They relate to, honestly, to the extent of processing nitrogen, the process controls on isotopes. But in dry areas, the nitrogen isotope values tend to be higher than they are in wet areas. And that's true on the landscape, like a low wet area at the base of a hill and the high top of the hill. There will be nitrogen isotope gradients in vegetation, largely driven by processes going on in soils that are inherited by the plants. For carbon, I talked yesterday about plants that do C4 photosynthesis, the tropical grasses, and they have high carbon isotope values in this range. And then the C3 plants, the trees and shrubs, in dry areas, they have higher values, and in wet areas, they have lower values. Again, due to kind of constraints on the photosynthetic rate and how that sorts isotopes. So that's the control. That's the kind of the isotopic landscape we're going to try to interpret lemurs on. And so I've plotted these gray ellipses, and they represent samples of bones of lemurs. So this is their bulk diet. The diet over the time period by which the bone was formed is bone collagen, protein in the bone. And each ellipse represents a different extinct lemur. And there's an example of one, Megalatopus, 85 kilos. It's one of those ellipses. I honestly can't remember which one. Okay. This paper was published in 2012, and my memory is not that good. I'll just use this, it's faster. That ellipse represents data from 3 or 4 species of surviving lemurs that were alive 900 years ago when all those big guys were around. And so you could imagine that this is, I was interested in whether these lemurs, the survivors, were competing for food and space with the extinct lemurs. And the answer is there's not much overlap, actually. They look like there's a bit of partitioning of resources, food and space, between the ones that are gone and the ones that survived. So a question would be like, after the big guys check out for whatever reason, do the survivors, do they have the capacity to expand their behavior and move into these open niches? This was just to show you the cute surviving lemurs. Okay, so not 85 kilos anymore, okay? And that yellow ellipse shows you the data for those same surviving species after the other animals have checked out. So there's like a subtle move of the ellipse, but it's not towards the isotopic habitat, isotopically defined habitat that was vacated. They've just kind of drifted maybe it got a little drier in Madagascar, and they just drifted towards a more arid adaptation. So no competitive release. If they were being held in that location by competition with the extinct lemurs, once the extinct lemurs were gone, didn't make a difference. They did not go into those niches. And then this is what they look like today. They are clearly, and I told you this, they're in the wet riparian corridors. So this makes sense that this is what's going on. But what this suggests is that in the past, they used that dry scrubland a lot more than they're using it today. So that is that could be a concern. It might suggest we should think about protecting the scrubland more because it's habitat that they have the capacity to use. Um, but I want to talk a little bit about what we think caused their extinction and what more we might learn about that from studying lemur fossils and fossils of other organisms. And so there's evidence for potentially evidence for increasing aridity over the time span of the extinction from 2000 to 500 years ago. Clearly humans arrived on the island and they definitely hunted these animals. Competition with domesticates, and then some combination, unholy combination of all these three things that, led to the demise of this cool fauna. And Brooke wasn't the only person to work on this. There's been people who followed up on her work, and she's followed up on it. And there was a paper in 2021 by Thomas Hickson, where he looked at the arrival of the domestic species in Southwest Madagascar. And this is a little complicated, this plot. Here are the arrivals, pigs, cows, sheep, and goats here, or shoats. No, these are the goats. And these plots have a little bit of error on them. They're basically, from three specimens, you can estimate, the most likely first arrival in Madagascar. Time is running from 2,000 years ago to today. And so, by 750 years ago, it's likely that there were pigs on the island. By 750 years ago, it's very likely based on these three dated samples of goats that they had arrived on the island. Similarly, he used statistics on his data and Brooks' data to think about the kind of the most likely last occurrence dates of various extinct animals. And Thomas looked at the giant elephant birds, two of the lemurs that Brooke looked at, sadly extinct tortoise, and the hippopotamus went extinct. And so he's got age estimates for when these animals go and when the domesticates arrive. And these histograms are overlapping with each other. So it is definitely possible that the animals were interacting with and affected by the arrival of the domesticates. So I'm going to show you these same figures again that I walked you through before, but I'm going to end by showing where the domesticates plot to think about not just are they there at the right time, but are they doing the right things to be trouble? And so there are the extinct lemurs, and Thomas measured a few more, actually quite a few more, of two of the species of extinct lemurs. So I'm showing his data plus Brooks' data with these new colored ellipses, but it's the same 5 species as before. I should have noted there's one C4 feeding lemur from back in the day, one grass eating lemur from back in the day, kind of cow lemur, I don't know. Here again are is the isotopic niche for those survivors, not before 900 years ago. Here's the isotopic niche for the survivors after the extinction, like no movement. And here are the isotopic niches for the different domesticates. And what you can see is that Ovis wasn't really competing with many of those extinct species except for this grass-eating lemur. Ovis would have been trouble for this grazing lemur. The cows, again, not terrible trouble for most of these species, but they do begin to overlap a little bit. The problem are the goats. The goats are definitely getting at the stuff that the extinct lemurs liked. And they're also getting at the stuff that the survivors liked, right? Now, whether they were dense enough to trouble these survivors is another question. But remember, this is where the survivors are today. They're not competing that much with goats. They found a way. They found a niche now that doesn't compete with the goats. And it may well be that they're not just in the forest to hide from people. They're in the forest to hide from goats or to find food that the goats can't get. So it's a really cool extension of this work that Brooke did, and she's on some papers with Thomas now, kind of thinking about this interaction between potential causes of extinction and species contraction, and really getting into the nitty-gritty of what's driving that and how adaptive these surviving species are. And they've got quite a bit of adaptive potential, but there's not that much riparian corridor. So, I mean, it may work, but your population numbers are going to be low. So there's some words here. No sign of competitive release. Niche breadth was reduced after the big lemurs went extinct. No lemurs went back into that space. The competitors definitely were in that space and could have been trouble for the extinct lemurs and the survivors. The modern lemurs somehow lived in the face of it for a while. They were living with domesticates, competing with domesticates, but now they're not. Now they're basically in these reserves, and that competition with goats may be a factor in their restriction today. And the end result of this is it's really critical to protect some area away from the rivers to see if we can let and maybe exclude goats and see if we can get lemurs to have a bit more habitat like they had 900 years ago. So that's lemur. I could stop here and take some questions. I should have is there a clock in here? Oh, there's a clock on my, so I'm about halfway through. Cool. Gabrielle. Yeah. Yeah. Yeah.
Like the circles are smaller, right? The ovals are smaller.
The ones that survive, the circles are smaller. The ovals are smaller.
Yeah.
The dashed lines.
So I'm just wondering about the variance. Like,
Yeah.
is that from sampling or is that?
It's a mix. Yeah. These are Bayesian ellipses. So they're 95. I think these are 95% confidence interval that the data are falling within that ellipse. And the fewer the samples you have, the ellipses can blow up. So some of it is sample size, but some of it is just real variation, I think.
So that dotted broader range?
The ones that are dotted are a couple of them, there were only 3 or 4 specimens, right? But some they had a robust number of specimens. So it's a mix of I would say that the first axis is a good representation of the location of the plots. The points, right? The second axis is going to explain less of that. Anyway, there are sample size limits here. There's not that many specimens. Yeah.
Base isotope composition hasn't changed over the last 1,000 years, and in particular, whether Haber-Bosch process and contamination.
Yeah. So probably not. The collagen would not be contaminated. If it was mineral, it would be more of a worry. If I was showing you data from bone mineral or tooth mineral, that can exchange with soil solutions. But collagen gets cleaned up, and so collagen's collagen. If I was showing you modern data, the modern data is probably, we have corrected it for the change in the composition of the atmosphere due to the fossil fuel burning. We have not corrected it for Haber-Bosch nitrogen deposition. And that's like highly variable globally, right? So it's hard, it can be very large or it can be small. But that's a good point that the carbon is corrected for anthropogenic effects. The nitrogen in that green ellipse is not. Okay, 2 more and then I'm gonna go to seals. Yeah, go ahead.
Eating similar plants, but just moving up higher in the feet, like escaping competition.
Yeah. Depends on how high a goat can climb. Yeah. I don't know. They can climb. Yeah. There are, but the isotop values drop as you go. In a dense canopy forest, the isotop values get lower as you go from the top of the canopy to the floor. The riparian corridor might have that effect. The spiny scrub is the bushes are spaced too far apart for that to matter. Yeah, and there's another. Yeah, go ahead.
know, you have other evidence that suggests that food was limiting?
I think, yeah, good question. I don't really know the answer to that. I haven't done a lot of work on the modern. If Brooke was standing here, she would just flow with information on this, but I'm not Brooke. So I don't, she has worked a bit on how these plants are defended against lemurs. They have spines and spikes that are defending them against lemur predation and lemur foraging. So we know that from a plant perspective, the lemurs matter. We don't know I don't know the reverse, how food limited they are, the other direction. This is really low rainfall area though, so there's not a lot of primary productivity in this area. So my guess is this is a tough place to be a lemur. It's a tough place to be a human, which is why this is the last place that they are surviving in Madagascar. These particular species are surviving in Madagascar. Okay, so now something completely different. Okay, so going to the Ross Sea coast of Antarctica, as far south as you can go on a boat. That's why Scott and Amundsen went there, and Amundsen came back and Scott didn't. And actually, we're gonna a lot of the work that I'm going to talk about is done on this coast. This is floating ice. It's an ice shelf, the Ross ice shelf. And we all know that global warming is much stronger at the poles than it is at mid latitudes, and there's a whole bunch of reasons for that. But we expect these animals to be experiencing some changes, strong changes, earlier than other organisms. Let's put it that way. And I'm going to show you the main cast of characters. Weddell seals, they live in the sea ice. They have very diverse diets. They eat benthic food, they eat pelagic food, and they maintain their ice holes by chewing them and gnawing them open, and their teeth are all worn down from doing that. They breed on the ice, and they dive in the water to get warm because the water's 4 degrees C, but the air is -20. So crab-eater seals also live in pack ice, maybe not big floating chunks of sea ice. They have these ridiculous teeth with hooks on them that they use, kind of like baleen whales use. They strain krill out of the water. They take a big mouthful of krill, and then they use their tongues to push the water out, and these hooks prevent the krill from flying out. And this is one of the most, biomass-wise, one of the most abundant surviving large mammals. 10 to 20 million of them, 3 times the size of a white-tailed deer. There's a lot of biomass of crab-eater seals in the world still, and that's because they eat almost at the bottom of the food chain, right? They don't eat fish. Well, today they don't eat a lot of fish. They eat organisms that are very low in trophic level. So there can be a lot of them. And then the other seal, the one that got my attention to this site, is the southern elephant seal, the largest seal on Earth today, bigger than its northern cousin. It's ice tolerant, but it doesn't currently occur in the Ross Sea in any abundance. And so I'm going to tell you that the Ross Sea is icier now than it was in the recent past. And what's the evidence for that? Well, there's geomorphic evidence. So this is the Ross Sea coast today. That's the actual coast. And this is a floating ice shelf, a sea ice shelf. It's not like this 300-meter thick giant Ross ice shelf, but it's basically it's sea ice that's frozen to the coast. And most of the time, even in the summer, there's tens of kilometers of ice from dry ground, this little bits of dry ground here, and then these are the dry valleys of Antarctica. But these little beaches here along the coast, there's tens of miles of ice from water to that dry land. The only place that's not true is up here in Terra Nova Bay, and I'll show you some data from Terra Nova Bay. There's a polynya up there. There's insanely strong winds blowing here, and it pushes the ice out off the coast. And the geomorphic evidence are beaches. there are all along the Ross Sea coast, there are raised beaches. And these beaches are formed by liquid water rolling boulders and gravel. And that's not a thing that happens on most of the Ross Sea coast today. Ice pushes rock around today on the Ross Sea coast, but you don't get water-generated beaches. These beaches are, the oldest beach here is about 7,000 years old. They're all isostatically uplifted due to the retreat of that floating ice shelf wasn't always floating. It used to be grounded. At the full glacial, it was pushed way out almost to the continental margin, and it pulled back about 7,000 years ago, and the land surface is popping up because it's been unloaded by kilometers of ice. The other sign that it was less icy on the Ross Sea coast is the presence of elephant seals. There's a few places where there are mummies of elephant seals. That's my colleague Brenda Hall, noted glaciologist Brenda Hall, being a seal biologist for a bit. We find the bodies of elephant seals on the beaches along the Ross Sea coast. But the main way we find elephant seals on the Ross Sea coast is as fur. Elephant seals molt their skin catastrophically. And we found elephant seal fur at all these sites along the Ross Sea coast and here on Ross Island by digging in the gravel, stealing spoons from McMurdo that we can use to scoop fur out of the dirt, and radiocarbon date this fur, look at ancient DNA in this fur, and hundreds of samples of elephant seal fur. There's been one elephant seal seen here at Terra Nova Bay, scaring the Italians at their base in recent years. And there was a tiny male haul out here for 10 individuals of males that got down here and shed their skin and then went back to New Zealand or wherever they're from. But really, this is not elephant seal land. The elephant seals were only seen at the places where these polynyas exist. This Terra Nova Bay polynya. There's another polynya here at the Ross Sea where the ice gets blown away a lot. And there's a little bit of liquid water. And so sometimes the elephant seals will come there, but they're really not there today. It's a paper when you see one, okay? You write a paper, a note, a zoological note. And where the elephant seals are today, they breed on these subantarctic islands, and they haul out to breed. The males are on shore for a few months. The females are on shore for 3 weeks to a month. And they come back, I don't know, 2 months later to molt their skin. Most of the time they're out in the ocean feeding. And there's a few spots in Antarctica on the continent or islands right off the continent where males will stay to molt their skin. They won't swim back to their natal islands, to the breeding islands. But most of the males go back to these islands. And Macquarie Island is the closest breeding colony to this Ross Sea coast where we're finding elephant seals. So we've radiocarbon dated a lot of elephant seals, and to figure out their time of first appearance and last appearance, they first show up about 6,000 years ago. They last are abundant about 1,000 years ago. They kind of drop out of the record 1,000 years. There's a few straggling specimens after 1,000 years ago, but they're pretty much gone 1,000 years ago. It's a little variable up and down the coast. There's peaks that may relate to warm periods versus cooler periods. I'm not gonna talk about that. There's other evidence of changes in the Ross Sea coast in the late Holocene in the last 1,000 years. These are nitrogen isotope data from penguin eggshell organics. So the penguins do breed along the coast here, and their eggshells have little bits of protein in them. And Steve Emsley and Bill Patterson measured that stuff, and they saw this weird, very distinct pattern where there's high nitrogen isotope values in the penguins, and then about 1,000 years ago, 750,000, they just drop catastrophically. Remember I said nitrogen is trophic level. So they interpreted this as, penguins are eating lots of fish, and then they get basically restricted to kind of krill diets here in the end of the Holocene. And they said that was because humans had killed off all the baleen whales. So the baleen whales eat krill too. And they're like, oh, well, penguins had a big competitor in the Ross Sea until industrial whaling took out the baleen whales. It's the krill hypothesis, the krill surplus hypothesis. The other thing that could go on in the Ross Sea is, so this is a food chain, starting with phytoplankton and all the way up to seals and Yeah, that's a seal eating a penguin. Okay, so and whales that they're killer whales in the Ross Sea as well. And so they've yeah, this is just to say that they interpreted the fish eating penguins versus krill eating penguins. So they interpret this record as a food web shift. But there are other ways for isotopes to change. There can be changes in the isotope composition of these phytoplankton at the bottom of the food chain. And to go fast, what I'm going to say is that when the water is more productive, when there's lots of photosynthesis going on, the values for these phytoplankton are higher. And when the water is not productive, when there's not that much photosynthesis going on, the values for the phytoplankton get lower. And I'm gonna skip the slide showing the experiments that demonstrate that to save your brains. So there could be shifts, that the stuff that they were seeing in the penguins might relate to some sort of baseline shift from more productive to less productive water. So what we did with the seals was we sampled lots of elephant seals, we sampled lots of crab eater seals and Weddell seals to ask if there was evidence. We know it was less icy, but how else was the Ross Sea different for 1,000 years ago? What was the fate of the elephant seal population? Why did it go away? And then, are there changes in the abundance or ecology of the animals that like it there today that might somehow relate to these shifts between more or less productive, or like baleen whales there and baleen whales gone, or more or less icy? So we're trying to look at how these animals, both what happened and how the animals respond to it. And so just, if you do field work in Antarctica, you got to show some pictures. So this is a photo taken not by me, from a helicopter looking out towards the ocean, flying over one of the dry valleys. We camped at this site. I can't remember the name of the site. And then we camped, near some of the glaciers up in the valley. And what we would do is we just walk all over the dry valleys and the beaches looking for dead mummies of seals. And we'd walk as far as we could walk from any one of these campsites, and then we'd helicopter different places. Then we'd walk as far as we could around those sites, and hope we could get back to our survival bags before the helicopters came back. If you turn the helicopter around, look the other way, and look up towards the continent, you see the continental glacier coming down into the dry valleys. But it doesn't make it into the dry valleys because it's so dry in this area that the continental glacier is sort of starved for water. But it's kind of like, I don't know, Game of Thrones. Like there's this wall of ice behind you. That plateau is 10,000 feet. In the other direction, we were looking at sea level. So the back of these valleys, yeah, there is a wall. There's a wall of ice, or a ramp up to a wall of ice. This is how the field work works. You get dropped by helicopter. You set up some tents. You hope that it's not cloudy, because if it's cloudy, then this little stream stops running. And then you got to go run up to the glacier and chip ice off for water, and that's challenging. We were looking at actually the breakdown series of some of the fossils. I was interested in taphonomy and how the fossils break down, like how long they last on the surface. I don't have time to talk about that. And we sampled many localities. This is a plot showing our data. So green is elephant seal just on the coast. The blue is Weddell seals. They do go inland. The black is leopard seals. And I didn't indicate that here, but the dots are here. And then the crab eater seals are in orange. And 400 crab eater seals found, 175 Weddell seals, 40 elephant seal bodies, couple hundred fur samples, and then 25 leopard seals were found. And what we did when we got there, we took samples for ancient DNA to ID the species and measure population size changes. The main team did radiocarbon dating, and then we did all the isotope chemistry stuff. I'm going to show you data from individual amino acids in seals, but I'm mostly going to show you bulk carbon and nitrogen data. So that bulk carbon and nitrogen data in the fossils is going to come from bones, either bone collagen or dentin collagen. I'm going to show a little bit of modern data that comes from blood or fur. And then for nitrogen, I'm going to show you data comparing amino acids that don't change in nitrogen isotope composition as you go up the food chain versus amino acids that do change in composition as you go up the food chain. And the difference between those two is a measure of trophic level that you can get on a single fossil. Because this is the bottom of the food chain, that's the top of the food chain. And kind of the way it works is that with each trophic step, there's a tiny increase in the nitrogen isotope value of the what we call source amino acids, but the trophic amino acids jump fast. So it's an internal trophic level monitor. So here are the bulk data, the bulk trends in nitrogen isotopes for the elephant seals. And what you see is this decline from 7,000 years ago, the oldest, 6,500 years ago, the oldest specimen we have. There's a subtle decline. Not really like what we saw with the penguins. Penguins were flat over that time period and they crash, right? But the elephant seals check out right when the crash begins. The penguin crash began. I'm not going to show you the carbon data because it's not important for the story. Nitrogen isotope values in the Weddell seals, not as far back. We can't find 7,000-year-old Weddell seals. They're pretty flat, and then they begin to drop. Actually, if you go to Scott's Camp and sample the dead seals from Scott's Camp from 1911, they've started to drop, and then these are the modern Weddell seals. So a subtle drop, but nothing like what's happening in the penguins. And the crab-eater seals, they're just all over the place. And the modern crab-eater seals, though, so there's big spread in the fossil crab-eater seals to potentially higher trophic levels. That would be a small fish-eating crab-eater seal and a big fish-eating crab-eater seal. And the modern crab-eater seals, all but one, just strain krill. And this crab-eater seal was hanging out with the Weddell seals, like by a hole in the ice, and it seems to have been confused. And it was just doing Weddell seal stuff. It was feeding benthically like the Weddell seals. So that's the bulk data, and there's information, about flex adaptability. The Weddell seals do seem to be able to shift with the environment if it's a baseline change or a change in the abundance of krill. The crab-eater seals seem to just be doing something different in the past. Now we're going to use that approach to measuring trophic level. So it's hard to do, so just a few samples for bulk data. And then I'm going to show you, these are the phenylalanine data. So that's the baseline for the system. And so baseline here for the crab-eater seals is pretty flat. Baseline for the Weddell seals shows a drop, but it starts earlier than the drop that you see in the bulk. And when you look at the source amino acids, which are showing you sort of the ones that respond to trophic level, you see that same drop occurring in the Weddell seals happening earlier. And you see sort of a bimodal distribution for the crab-eater seals. Some of them are really high and some of them are lower. And I wish I'd chosen different colors. Okay, I'm sorry. But these show the patterns, right? You see this drop, parallel drops in the source amino acids and in the trophic amino acids. That means the trophic level stayed the same. So they responded to something happening in the environment, but they didn't change their trophic level. It's got to be a baseline shift, actually. And the crab-eater seals, like some of them are low trophic level, like today, and some of them are high trophic level. Some of them were eating fish back in the day. So not something we're seeing. I'm going to shift to talking about the elephant seals now. So remember, these are the ice seals that like it there today. And now I'm going to talk a little bit more about what's going on with the elephant seals that left 1,000 years ago. So this is data from elephant seals all over the world, mostly collected by marine mammal biologists from all these different potential rookeries. And there's this clear array between high carbon and nitrogen isotope values in these lower latitude regions, and low carbon and nitrogen isotope values as you get into these unproductive waters around Antarctica. Remember, I mentioned that when productivity is lower, when there's more nutrients in the water, the isotope values are lower, and you're seeing that reflected in the animals that feed in those waters. And in the let's say the almost temperate latitude waters, all the nutrients are sucked out of the surface water. And the nitrogen and carbon isotope values get higher. So this array is just a productivity array. And you can see that, I'm not going to dwell on this, but these are ocean cruises that have mapped the nitrogen and carbon isotope value of the surface oceans compared to a model. And that drop in productivity and the drop in isotope values is obvious in modern systems today. So there are, I mentioned these places where the males molt their skin on land. And here's one, and here's two others. And when you look at the data from, in particular, these sites that are on the shelf of Antarctica, they have high nitrogen values, but low carbon isotope values. So that's kind of weird, like what's going on? And these sites are places where there are polynyas. Sorry, I need to. These sites are places where there are polynyas, where the wind is blowing the ice away. And when you go in and you measure productivity in those polynyas, it actually can be really high, just right up next to the ocean, right up next to the shore in a polynya. The isotope values can get high. Sorry, the productivity can get high. And that's because there's dust blowing off the continent and there's light getting into the water. And the water's kind of stratified, so it's clear. So there's lots of photosynthesis going on in the polynyas. And people have taken boats and gone down and sampled the water and measured the isotope composition in these polynyas. And yeah, low productivity water here, that's this pale blue color. But when you get into the polynyas, you see these red colors. That's because the nitrogen isotope values are climbing because the water, it's very productive in those regions. So there are these little hotspots of productivity next to the coast of Antarctica right now. But most of the water around the continent is unproductive. And the Ross Sea is one of the unproductive sites, although these people thoughtfully didn't include the data, these open circles. They went there and sampled, but they didn't put the data in the paper. So I'm going to skip the penguins. Well, I won't skip the penguins. So people, Emsley and Patterson and others went back and used this trophic level approach, source versus trophic amino acid, to look at the penguins. And what they saw was this very subtle decrease, and yeah, maybe a tiny drop in trophic level, but this big pattern, the krill surplus is gone. That hypothesis died in 2023. There's some subtle baseline shift in the system and some change in trophic level. Well, there's there's a combination of baseline shift and a subtle change in trophic level. This is trophic position here, not isotope value. So like a half a trophic level drop in the penguins, but not 2 or 3 trophic levels, the way they interpreted it in 2012. So some evidence that there's been a shift in the baseline and that it was more productive in the Ross Sea in the past. And I'm going to show you some evidence just about species interaction. So crab-eater seals, mostly fossil crab-eater seals back in the day, going to much higher, with some individuals going to much higher trophic levels, if nitrogen is a trophic level, and much higher trophic levels than today. These are the Weddell seals today and in the past. And they are at higher trophic level. And slightly, we already said that their nitrogen values have shifted a little bit, but that's due to that's really these samples right here that are showing that nitrogen shift. The bulk of the Holocene samples are in here. So there was some competitive interaction between Weddell seals and crab-eaters, at least the fish-eating crab-eater seals in the Ross Sea. And the elephant seals, a lot of them were feeding out in the circumpolar currents, but some of them were feeding closer to shore, and they were also competing in this productive water with the Weddell seals and the crab-eater seals. So I'm going to skip the genetics, sorry, other than to say we know from the red are the Victoria Land, the Ross Sea genotypes. We know that the Ross Sea colony was founded by Macquarie. It evolved independently of Macquarie. There are now some genotypes from extinct colonies here in Antarctica and New Zealand, and there's some shared genotypes with the Ross Sea. between these other extinct colonies, but none of them make it into the recent. These are all empty haplotypes. So these are mitochondrial haplotypes. The size of the circle represents how many individuals are in the haplotype. And these are cleaned out extinct haplotypes that were present in the Ross Sea for thousands of years. So the Ross Sea population appeared, it evolved independently of the rest of the elephant seals around Antarctica, and then it went extinct and didn't leave survivors. Using skyline plots, we can determine how many elephant seals there were. Effective breeding population of about 250,000 elephant seals at their peak, and down to 40,000 elephant seals today. So that's a lot. That's a lot of elephant seal. Okay, that was a really different ecosystem supported by a more productive Ross Sea, because the Ross Sea was less icy. The Ross Sea was essentially a giant productive polynya. That's our hypothesis. For most of the Holocene, it was like those little hot spots along the coast today. And the elephant seals kind of respond as you'd expect. When it's productive, they appear when it gets less icy, and then they're super productive and feeding down there. And then once it ices up, it becomes less productive and they crash. The same approach is applied to Weddell seals. The Weddell seals, they notice the disappearance of the ice sheet, but they're pretty much, this is flat for a skyline plot. They're not showing much change once the ice disappears. In the Holocene, their population sizes are about stable. And the crab-eater seals, which you would think would respond to the disappearance of sea ice for much of the Holocene, they also just have this linear increase in population size over the course of the late glacial into the Holocene. So they don't seem to notice the disappearance of the ice. It's a really interesting response, different population response in these three seals. And they don't seem to be necessarily noticing each other. Like there's not a response of the Weddell seals necessarily to this giant elephant seal population. So intriguing things. Coming back to kind of summing it up, the Ross Sea was less icy and more productive prior to 1,000 years ago. That's what some of the isotope data are suggesting. All the species exploited that less icy productive shelf habitat. I should have said this. The Ross Sea is the largest shelf habitat in Antarctica. And for it to be open and available to be foraged in and productive is a big deal for productivity around the Antarctic continent. And that it's closed off now and has been closed off for the last 1,000 years is important. Elephant seals exploited that habitat as soon as it opened, and they stayed there until it closed. The crab-eater seals had surprising diet breadth, and they had that even after it became less productive. It's just the modern crab-eater seals that don't seem to eat fish. And then the Weddell seals had the same diet, at least the same trophic level, going from past into present, but they just, when the baseline shifted, they just kind of followed it down. And the species interactions among these species is super interesting. So, what this can say about adaptability. Okay, so the elephant seals are extremely adaptable in where they breed and how soon they find breeding colonies and how they exploit the habitat. They seem like they might be able to deal with a less icy Ross Sea in the future. The crab-eater seals had surprising adaptability, and they don't seem to be sensitive to this de-icing, which, this de-iced Ross Sea. They're not sensitive to it. They were still there, and their population sizes were increasing linearly through it. So this climate change, they just don't seem to care about. And the Weddell seals, low adaptability, they just keep doing the same thing through time, but it doesn't seem to be affecting, at least currently doesn't seem to be affecting their, they don't seem to be sensitive to these changes. They just keep doing the same thing as it's more and less icy. So we're beginning to get some sense of how these species might respond to a future state where there's not much ice in the Ross Sea. And the species interactions are going to be very important. So I'll just end this part by saying, thousands of seals made poor decisions and wandered inland and turned themselves into mummies, or they died on the beaches. And that's, bad for them, but great for us. We see, surprising flexibility in some of these animals. Not things that we would imagine necessarily from what they've done over the period of observation. And the future thing to remember is that, a less icy early Holocene might not be the same ocean as a less icy Anthropocene, right? The way that ocean worked from 7,000 years ago to 1,000 years ago may not be the way that it works if we heat the planet up and melt all the ice off it again. So it's cool to know they can be flexible and adaptable, but we're going to send them into a world that hasn't existed for millions and millions of years. So gives me some comfort to know how flexible they are, but I don't sleep especially well when thinking about this. So I'll stop there. So not lemurs, but questions about seals? Yeah, Don, and then in the way back.
You have last play that with a lot differently between 7,000 and 1,000.
Yeah.
Today, what's the ultimate driver of having? Why did I stop last?
Yeah. Okay. So I'm going to say this thing that I've said before, and I think the Ice Age was starting 1,000 years ago. I think the Holocene was ending, and the first place we saw it end was beginning to end, and we saw it here. If you look at ice core data, the elephant seals, and you look at temperature on, from ice core data, the elephant seals appear at a particular delta 18 value, and they disappear at exactly the same delta 18 value. The temperature has been dropping over the last 4,000, 5,000 years in Antarctica, and they disappear at exactly the same delta 18 value on that ice core curve. So I think there was just a threshold that causes the Ross Sea to ice up. It's actually probably an ocean current threshold, not an air temperature threshold. Orbital forcing that gets warm circumpolar deep water up onto the shelf. So that warm water is 4 degrees C rather than 1 degree C. And that warm water probably was melting the sea ice from the bottom up. And that warm water fell off the shelf about 1,000 years ago. The places where it still gets up on the shelf occasionally today, and it does help create the polynyas. So I think it was actually an ocean, orbitally driven ocean circulation warming. But the air carries some record of it. The ice cores carry some record of it, not the proximate driver, but the ultimate driver. Yeah. But that's the oceanography part. Yeah. So yeah, in the way back, there was a question. Yep.
So, what I gathered, the trophic levels don't seem to have affected much of anything except for elephants.
Yeah, the trophic level changes, they just didn't happen in the Weddell seals. The crab-eater seals do seem to change their trophic levels a lot. And the elephant seals really were just changing the places where they were feeding. Their trophic level didn't change. I didn't show you trophic level data for them, but their trophic level doesn't seem to change that much. They're a bit like the Weddell seals, except they're moving where they feed. So the elephant seals move in and then they move out. The Weddell seals stay in place eating the same thing, and the crab-eater seals stay in place eating different things. So that would be the quickest take-home message from the blizzard of figures I showed you. Gab? Oh, genetics. Yeah.
So different seal behave more varied, more less varied, or
Right.
So what about their genetics? Are they more, if you looked at the populations, would they be more similar, or are they all,
I don't know the answer to this because I haven't seen the global population data for Weddell seals or crab-eater seals. I've only seen the global data for the elephant seals. And, over the course of the time that I've been working on this project, it's been like 15 years, What used to be just mitochondrial haplotypes. Now it's nuclear, right? Now it used to be a segment of the mitochondria. Now it's complete mitochondrial genomes and some nuclear DNA that can tell you about function. So the genetic data keeps getting larger and larger and larger, but that means you have to go back and resample over and over and over again. And the elephant seal people have been the most excited about that because the elephant seals honestly mostly live at lower latitude, so they're easier to get to, right? It's really hard to catch a crab eater seal. My colleague Dan Costa, went down and spent like 3 weeks, 4 weeks down there, and they live on the ice, the pack ice, floating hunks of pack ice. You got to jump on the pack ice, subdue an animal that's 3 times the mass of a deer. Don't let it die, or you're never going to go back, and you might lose your job. Don't let it die, and don't have anybody die on your team. So 3 weeks they caught 12 or 6. I don't know. They can't. And one reason that we may not be seeing fish eating is that we pretty much only catch the adults. And maybe the adults just eat krill, but the babies eat more fish. And we never catch a baby and turn it. The way you know what's inside the seal is they do what's called gastric lavage. You basically put a tube in their mouth, and then you put water in the tube and it blows. You force them to vomit basically into a bucket. And so it's really hard to catch a baby crab eater seal to do that to them. And I should have said all but one of our crab eater seals are young of the year. None of them made it to a year old, with one exception. So it might be we're just looking at the behavior of juveniles that has not been seen because people can't catch the baby crab eater seals. So yeah, there's observational constraints. I'll just put it that way. Yeah. Yeah, Tammy. Yeah.
The ones that go.
Yeah.
So.
Yeah. So no, that's what I'm saying. I think the crab eater seal mummies are a very skewed population. They're young of the year, and they're not. We have one adult crab eater seal mummy, and that adult would have fallen right in the middle of the modern distribution in isotopic space. Everybody else is an animal that so what the crab eater seals do today is they breed on the pack ice, and the pack ice retreats towards the continent, and they follow the pack ice in, and eventually they're over the Ross Sea shelf, because the pack ice is way out past the continental margins in the middle of winter, and they follow it in, and then they give birth when they're kind of close to the continental margin, and then as the pack ice, builds again, the seals follow it back out. So when there's no ice in the Ross Sea, they must be breeding on the beaches. And these crab eater seals must, 1 out of 1,000 or 1 a year, must make just the wrong choice when it's time to follow the pack ice out. And they wake up in the morning, and there was open pack ice when they went to sleep at night, and they wake up in the morning, the ocean's frozen for 10 kilometers out, and they've got to walk. They've got to walk to find water, and some of them make the wrong call. This is the first time they've ever done it, right? They've got to find open water. Mom's long gone, okay? Like crab eater seals wean their young at 3 weeks, so mom has left them. They're on their own, so they've got to figure out how to find open water, and just we have dated from the dating of the samples. Essentially, we have 1 seal per year. We have so many dates, right? That roughly we have a distribution of, so 1 seal per year could make a bad choice, and it would create the mummy distribution that you saw when you were in the dry valleys. The Waddell seals are different, and I don't have there are adult Waddell seals, right? And there are baby Waddell seals, but for the crab eater seals, it's all young of the year. Okay, I better stop here. People got to go. I have to go. Okay, we have a 5:45 reservation. So anyway, thank you so much again.